The overall goal of this procedure is to optically report the membrane potential changes with a genetically encoded voltage indicator.
This method will describe the strengths and weaknesses of imaging with different genetically encoded fluorescent probes of voltage. For instance, FRET-based probes will offer the advantage of ratiometric imaging, but will give a lower signal.
The main advantage of this technique is that we can visualize the neuronal activity in real time.
Generally, individuals new to this method will struggle because the signal-to-noise ratio is not very large and there are several sources of noise and multiple steps that can go wrong. New users often confuse any fluorescence changes as the real signals.
Visual demonstration of this method is critical to ascertain the validity of the probes being used and what it actually indicates.
For the setup, an image splitter is installed between the slow and fast CCD cameras for the imaging of FRET-based GEVI. Before the experiment, insert a filter cube with a dichroic mirror and 2 emission filters in the image splitter. Remove this second filter cube when imaging a GEVI with a single fluorescent protein. To begin the experiment, locate a healthy HEK293 cell that shows strong localized membrane fluorescence compared to the internal fluorescence, and avoid patching circular cells as they are either dividing or dying. Then, apply a test pulse to